All right, this will finish out our third video in our lecture on cell signaling for AP Bio Unit 4. This section is going to cover some of the more common and important receptors in this process, okay? And in eukaryotes, there's two big categories of these, and they are G protein coupled receptors, or GPCRs, and ion channels, which you've probably, that one's probably a lot easier for you to visualize, okay? So we're going to start with the one you probably haven't heard of yet, and that's the G protein, okay? So you have this GPCR process in the cell membrane here, okay? And this is a receptor, and you can kind of see that red thing coming in there. What would a biologist call that red thing? They'd call it a ligand. Yeah, very good. Thanks for jumping in there, everybody. Okay. So this is the biggest category of receptors in eukaryotes, and you can kind of see that it kind of like swirls up and down through the membrane, okay? And so a lot of different things are happening. We're probably seeing some amplification and some transduction going on in there. This process is really important in animal senses. So their sensory system, like your sense of touch, but also things like sensing what the environment is like chemically. So the internal environment of your body. So what we have, so this G protein thing, when we look at evolution, we see like the development of ATP being used widely across almost all every organism that I don't think there's anything that doesn't use ATP. But we think earlier in biological evolution, there may have been another molecule very similar to ATP called GTP. So GTP, super similar. It's got the 3 phosphates. So the G, which I can't remember what it stands for right now, is structurally very similar. You've got this little group with 3 phosphates on it. So if there's GTP, there's also GDP, just like there's ADP. Kind of overpronouncing so you can, clearly hear me on the video. All right. So what happens, okay? There's a GPCR enzyme and a G protein that stay inactive until the ligand binds to this extracellular site. Okay, so this picture right here, pay attention to this, especially look for that little green oval there. Off to the side of that little green oval is sort of like a site that kind of looks like a half circle, okay? So watch that half circle as I advance through the slides, okay? So this is going to stay inactive until the ligand hit. That right there is pretty easy stuff, okay? You can see we've got an enzyme floating in the middle of the bilayer. So let's take a look at what happens. All right, so now it's been activated. Okay, I really want you to look at what happened between the two pictures. I'm going to back up, look at where the active G protein is. Okay, right now it's just kind of waiting on the inner membrane. Okay, it's got GDP on it. When we attach the ligand, the things change. Okay, what's changing here? Okay, the G protein binds to this big complex in the membrane. All right, so you see here the ligand has attached, The G protein is also attaching internally. Okay? Why did that happen? There's some sort of change in shape that allows it to bond with that site. When it bonds, this GDP, that's a diphosphate, becomes GTP. Okay, so we went from a di to a tri. All right, let's take a look. After that, we have the activated section. So this whole complex together attaches to an enzyme. And that activated enzyme is the next step in the signal transduction pathway. Okay? And once that enzyme is altered, that is going to activate something specific internal to the cell. Now, I want to emphasize something here. There are multiple GPCRs. This doesn't just happen like one way. Those are all going to have their own individual ligands. And so we have one ligand that comes in and activates this enzyme, and then the pathway is really going to sort of activate, amplify, and take off. Okay, let's go back and just quickly review that. Okay, here it is inactivated. The G protein, where is that? That's internal. What's it doing? It's just sort of waiting. It's got GDP on it, so it's in sort of that lower energy state. Once the ligand attaches, that GDP gets phosphorylated into GTP. Now think about this. That's going to change the state of this again, so that's going to release the G protein, and it's going to go to that enzyme that really starts amplifying and getting the pathway activated. So lots of complexity here. Again, new stuff you probably haven't seen before.
So the GPCRs intracellular?
Well, the GPCR it's a membrane protein. Okay, so
it can be either.
It's going to be a portion of it is exposed to the outside set of the cell, and a portion of it is.
Yeah. Okay, that makes sense.
Okay.
So I saw that the binding occurs on the extracellular side, but you're talking about
the ligand.
Yeah. Okay.
Good, very good. That's a very good distinction. Like, your question tells me that you're kind of thinking in the way that the AP material is themed. What's happening? Where's it at? Okay. So when anytime there's a process, they almost always are asking you to break down the what, the where, and the order. Okay. So that question, I loved it. Okay. All right.
So it happens on both sides?
Yeah. Okay. So the ligand is going to happen extracellular, and the G protein is going to happen intracellular. The enzyme is going to activate things that are intracellular.
Okay, thank you.
Good. All right, great. All right, let's go to the second one. We're almost done. It's Friday. Let's wrap this up. All right. Ligand-gated ion channels, probably something you've seen but never named quite this way. Just like the G protein, this one is located in the membrane, okay? These are super important to the nervous system and propagating a signal through a nerve. Okay, so what's happening? Receptors are going to be gates for ions, okay? And when The signaling ligand binds to the gate. It's going to either open or close, which is going to allow very quick diffusion of very specific ions, usually potassium and sodium. And these are really neat because once they get set off, they really take off. They are kind of like nerves are kind of like a vending machine. You know, if you walk up to a vending machine and the snack you want is $1, you put a quarter in, nothing happens. 50 cents, nothing happens. It has to reach a certain threshold for these to begin. But once they do begin, they really take off. All right, let's look a little bit more at these. Okay, so in this example, we have a neurotransmitter that's going to be the ligand. In this case, that ACH, which you see the little red triangle, ACH, that's your ligand, okay? And you have, in this case, there's multiple ions involved here. You've got sodium, calcium, and potassium, okay? Part of this is inside the cell, part of this is outside the cell. And when this is activated, sodium ions will rush in because they're on a higher concentration on the outside, and potassium ions will rush out, which you can kind of see. See the arrows on the right side, the white arrows that kind of bracket the picture. So this is passive transport, but for this to get here, this stuff had to be all actively pumped out. Nerves can do this really, really fast, okay? Nerves use a lot of energy because they're constantly having to reset to this state, okay? Pound for pound, if I had a pound of muscle at full metabolic output, and then a pound of nervous tissue at full metabolic output, nervous tissue will burn tons of energy compared to muscle. When we think of fitness, we think of like moving our muscles, but nervous tissue is the hungriest tissue in the body for ATP and oxygen. So, you know, if you're going into shock, your body will do everything it can to keep your brain alive. First thing it's going to do is start shutting down muscles. That's why you get all pale, because it's pushing blood to your brain, okay? So just, it's interesting that we don't think of like a nerve as doing anything, but when we get down to the membrane side, this is all the passive side of it, but this has to be constantly reset, so nerves use a ton of energy. All we see here is very, very simple stuff. A neurotransmitter, ligand, opens up a channel. Ions rush through from high to low concentration, but they do have to be reset with active transport. That's what's not shown here. All right, and in this, I'm really kind of getting into that because my physiology background's coming out. But listen, what you need to concentrate on is that a neurotransmitter opens a pathway high to low. That's, in a nutshell, boiled down to the most basic. Okay, that was my last slide. Let me go ahead and stop that recording, and we'll talk a bit.